All right. Well. Welcome everyone that's joining us on Zoom. My name's Mac. I'm co-hosting this really exciting new seminar with Joe Lizier and Ben Fulcher. We're at the University of Sydney. We're in different groups. So I'm situated in medicine, Joe's over in engineering, and Ben's in physics. But we share a common interest in a kind of systems neuroscience perspective and complexity. So what we're trying to do to kind of sort of soften the edges of the pandemic is to invite people that whose work we really like, that we think sits at that really interesting intersection between these different fields, to come and talk and share an enthusiasm for a kind of new perspective that's emerging that intersects a number of different fields to try to develop a better appreciation of the kind of weird and wonderful shape of the brain. Formally kicking off our seminar this week is someone who we were absolutely delighted said yes to our request. Olaf, you do not really require any kind of an introduction, but all I'll say is that this man started out a love affair with network neuroscience for me. Reading his book, Networks on the Brain, when I was a grad student, just really opened up this whole new vista for how to think about a system and to take the neuroimaging results that we were getting and put them into a context that had a biological, realistic basis. And for that, I'll be eternally grateful. So I'm really excited to have you here today, Olaf, and we look forward to hearing you talk.
Thank you very much. It's really a pleasure to be talking with you, and I wish I was there, but it's not possible. And in this way, at least we get to have a conversation. I look forward to discussion afterwards also. So let me share my screen here and get the show on the road, as it were. Right. Okay. So. Great. This talk is in some ways appropriate for this seminar series that you're starting up, in part because, of course, it's all about complex systems and brain, the intersection of the two. I've been spending quite a bit of time in that intersection over the last 2 or 3 decades, and part of this talk will be survey and some old material, but I try to put a new spin on things and maybe come up with a few take-home messages, things that I've clarified in my own mind about the older work that are worth sharing. And I would like to then, at the end, hopefully the last 15-20 minutes, talk with you about a line of work that we've just started here in the lab and with colleagues here at IU the last couple of years that I'm very excited about. So a few words about network neuroscience. Of course, networks do not need an introduction anymore in neuroscience. They are ubiquitous, they're everywhere, with model organisms, the elegans, the rodent brain, mouse and rat, non-human primate, and of course in the human brain, represented through functional and structural connectivity. I always remind myself at this point that, this was not always so. And when I first got started working with networks and graphs back in the early 1990s, there literally was almost nobody interested in this line of work. It was a losing cause in some ways to invest in. But for some reason, they have taken off. And I think there's good reason for why they have taken off over the last 10 or 15 years. Part of the reason is that we now have a lot of data available to us that is very well suited for network approaches. Data coming to us from small scales, such as interacting molecules, all the way to large scales, interacting individuals in social groups. For the most part today, I'll stay in the middle part of this diagram, in that broad swath of time and spatial scales that are devoted to brain networks, connectome networks, functional and structural networks of the brain with model organisms, circuits, but also whole brain imaging. A lot of data comes at us. I remember a time when we had almost no data to work with. This was not such a long time ago, but now we are literally inundated with data. And networks are a very good, I think, very suitable framework for us to utilize, trying to make sense of these data. So part of what's driving our field, I think, is the fact that there's confluence of molecular, anatomical, functional, and behavioral data onto a common framework. Network analysis, to me, is not just a bunch of, MATLAB toolbox functions or a cute way of representing data. It's also a theoretical framework for understanding how the brain really works. So when I talk about networks, I talk about the networks as the real world systems. Networks are actually out there. They're not just in our imagination. I don't think it's just a metaphor, an analogy, but it is really how complex systems organize in the real world and across different scales. Network analysis tools are ubiquitous now. I don't think I have to explain a whole lot about it here, but a few of them stand out and will be referenced later on. The notion of communication through paths, sequences of edges, traveling through a network from one place to another. Modularity, the decomposition of a larger network into a set of clusters that are densely interconnected internally and weakly connected amongst each other, and the notion of a hub region or hub node that connects different clusters to each other. These are key concepts in our field, very widely applied. I'll have a few words to say about them and what I think is important about them in contemporary applications. So, few words on human connectomics. When I first got interested in the brain, which in my case goes back all the way to high school, the only way we really knew about how the human brain was structured was from postmortem dissection, histology, tissue analysis in sort of the classic anatomical manner. A picture here, a photograph actually of a microdissected human brain from a famous atlas from the 1950s. You're seeing the white matter pathways literally going through the interior of the cortex and connecting distant brain regions to each other. We did not have access to non-invasive methodology until about 20 years ago, when diffusion imaging and spectrography burst on the scene. As you all know, these are not direct observations of axons. These are inferred trajectories of white matter paths based on a complex diffusion signal that we can get from the MR scanner. So there's a lot of statistics and computational modeling going into a diagram like this. I want to emphasize this. It's not direct observation. It is a statistical, it's an inference. And there's all such things. There are false positives and false negatives. And while the field has been, going through various, stages of reckoning, I would say, with reality and with errors and biases in our data, for me, the glass is half full. We finally have a tool that's getting better all the time that allows us to look at brain anatomy in behaving humans across large cohorts, where we can relate variations in structure to individual differences in behavior and cognition. This has been a real breakthrough, a game changer, really, in our field. And I remember when this technology first became available, what a real big difference it made to our work. Working with Patrick Hagman at the time, we constructed a first connection matrix. This had not been done before, really, in the human, and we represented it as an adjacency matrix, as shown here, with weights indicated by color and presence or absence of connections indicated as well. This diagram here sort of it's a network map. It's a lookup table of what's connected to what. Each of the rows and columns corresponds to one patch of the cerebral cortex, left and right hemisphere. And we published our first paper with a grand total of 5 individual subjects. This is, of course, something that would not happen today anymore. Unless you have 500, you might as well not try. But we had 5 subjects back then, and this was our first attempt at characterizing network architecture, structural network architecture in the cerebral cortex. Interestingly, despite the fact that we had only five individuals in our first study, a lot of the things that we came up with at the time, a short little bullet list up here, really have held up over the years and have been, found again and again in different cohorts with different scanners and different processing algorithms. Just to go through these real quick, every cortical patch, every cortical region has a unique set of connections that travel from it to other regions, and we call that a connectivity fingerprint, a term introduced by Passingham and Olaf in 2002. Very useful term, I think. Why do we care about connectivity fingerprints? Because our connections in a network really are highly informative as to what kind of functional competence we have, how we relate to the rest of the network. That is through our connections, and characterizing connectivity fingerprints has actually become a very profitable exercise in network neuroscience. Brain regions do not all have the same number of connections. Instead, a few of them have more than the rest. So they are broad degree distribution, heavy-tailed, perhaps exponential, short path length and high clustering. In the cortex, at least, have been shown again and again. The hallmarks of small world architecture, modular architecture interlinked by hubs, and then hub regions that are also connected amongst each other into a structural core, a clique, or a rich club, as we call it more recently. This last property, just a few words about it. Why is it important? Why might it be important? Here we have a schematic network on the left with a few communities highlighted. They're densely connected internally, but not so much between. The critical nodes that connect them to each other are indicated in black. Those are the so-computative hub nodes. If I just add a few more connections between them in the middle of this diagram, you can intuitively see that now I can share information among these highly connected hub nodes, which are internally connected to a single community predominantly. I can now share information quite liberally and quite quickly and efficiently between them. That recommends this type of architecture, perhaps, for the type of processing that we might imagine goes on in a nervous system, where information integration, as many have postulated, might be a very important criterion for how we judge whether a brain is connected well or not. So that property, just abstractly from theoretical, first principles, might be an interesting one to encounter, and indeed we find it working with Martin Vandenheuvel a long time ago now. In our very first cohort, small cohort of individuals, we found this critical bump in the curve, which is the excess of connections between highly connected, high degree parts of the network, the excess of connections among them over a null model. These nodes are very widely dispersed across the brain, involving midline structures such as the anterior cingulate, the posterior parietal cortex, but also structures such as the medial temporal lobe and the insula. If you want to travel from place A to place B in such a brain network, and you take the shortest path to do so, we can argue later on whether that's a good approximation of how communication takes place in the brain. But for now, let's say we take the shortest path and we go from any place A to any place B. A very large number of these paths have to access some part of this relatively restricted infrastructure, the rich club, which only accounts for about 10% of the brain's nodes and connections. And yet close to 60 to 90% of all paths have to go through it. And there's clinical connotations here, potential for clinical studies to look at rich club damage, whether it has effects in clinical populations on network efficiency and communication processes. For me, very interesting, I'm working with John Beggs here at IU and also with Ben Dunn in Göttingen. Next slide, and I'll show in a moment. We've been looking more and more in cellular data, and working with John Beggs, who works with microelectrode arrays in culture, in vitro. A few years ago, we found that rich club organization was actually present also in those cultured in vitro situations in hippocampal slices, but also in dissociated cultures. And connections here, to be clear, refer to essentially effective connectivity, transfer entropy links between neurons that relate to information flow, perhaps more so than anatomy. Nevertheless, if we do look at highly connected nodes in such a network, and we ask a question whether they are more densely connected to each other than expected by chance, we find that is the case. To me, that was surprising. I did not expect to see rich club organization at the cellular scale, at a length scale of under 1 millimeter. We have since started working with Ben Dunn in Göttingen, who is a monkey neurophysiologist, to look at in his data, which involve multi-electrode recordings from 3 different cortical regions in a behaving macaque monkey. We've been looking at Edge-Centric organization in those preparations. He already published a very nice paper in 2016 showing that Edge-Centric organization is present, that Edge-Centric links and nodes span multiple areas and involve largely beta synchrony neurons that are synchronized in the beta frequency range, which is interesting from the point of view of information transfer, information sharing, and integration. Now we're looking at these data with Ben in great detail currently, time-dependent functional connectivity, time-dependent effective connectivity, Edge-Centric organization in relation to task, and finding interesting things, although it's too early to talk about what these results are showing us. It's work in progress that I'm very excited about. A few words now about comparative connectomics. Why? Because in some ways that is how I got started, how the field got started, looking at data sets from macaque and a few other species in the 1990s and early 2000s, work that I very fond of, still remembering working with my late colleague and friend Olaf. Really, we had very little data, and the data that you see here, this matrix of 47 brain regions and their directed binary projections, was almost all we had in the early 2000s. We threw everything at it that we could to understand the network organization of this very rudimentary, very simple binary macaque cortical matrix, and came up with a few concepts at the time. One of the things that we've looked at a few years later was Edge-Centric organization once again, and modularity in a larger collection of macaque connection reports, 240 brain regions. We did so because we had discovered Edge-Centric organization first in human imaging data, and as people who are familiar with the potential drawbacks of imaging, especially diffusion imaging and tractography, we were curious to see if we could find this type of organization in a more invasive methodology, such as track tracing or perhaps even single neuron reconstructions. And the answer is we found it, and it involves regions that are mapping broadly onto those that we see in the human as well, prefrontal regions, parietal, cingulate regions, orbitofrontal regions, temporal regions, etc. And Edge-Centric members of the Edge-Centric, highly connected regions that are also connected to each other quite densely, are distributed across the cortical surface, which highlights the fact that the Edge-Centric is in fact a costly adaptation. It's something that soaks up quite a bit of wiring cost in the primate brain, at least, as it's been shown. This is true even in C. elegans, and this has led me and Bullmore to propose a sort of an economic model of trade-offs to do with efficiency and cost, to which I'll come at the end of this section. A few other model organisms, just very briefly here. Drosophila, for which we now possess a much more fine-grained electron microscopic reconstruction of single neuron connectivity. Anyway, some years ago, we did a sort of, mesoscale reconstruction effort, network effort, together with Anshin Xiang's group in Taiwan, and found modular organization and Edge-Centric as well. In the mouse, this has been looked at over the years by several people, especially in data set coming from the Allen Institute. And again, modularity and Edge-Centric organization are present. And then in the rat, where I've been working for several years now with Larry Swanson, who is, really one of the world's leading anatomists and has a encyclopedic knowledge of the rat nervous system. And over the years, last 5-6 years, we have worked intensively, working our way through various subdivisions of the rat nervous system. Shown here from a relatively early part of our endeavor is the rat cerebral cortex. What goes into the construction of a matrix like this is published literature, histological reports on, regions that are connected to each other that have been ascertained usually with histological track tracing methodology. And the way Larry compiles these is literally going through all the articles that are published on any anatomical subdivision and collating the information in great detail. So every entry in this matrix quite often has multiple papers behind it, multiple reports of connectivity being present. It's a directed matrix. It's a weighted matrix too, because there is an estimate of the connection, of the magnitude of the projection on an ordinal scale at least. And it's all of the data sets that Larry has compiled for all these different subdivisions of the rat nervous system are quite full, in the sense that there are very few locations for which we have no information at all. So the fill ratio of these matrices is actually 90–95%. It's a fairly complete, very detailed type of data. And I've been privileged to work with Larry on this, and on very, I'm sort of doing the network stuff, and I'm always amazed how much it tells a person like him about rat brain organization, the sorts of things that we can do with such data sets. So one thing we can do is we can decompose it into modules and look for group membership and relate them to functional specializations. That works quite well. And as we encountered different subdivisions, differences have become apparent. So not every network, for instance, not every subdivision of the rat nervous system has small world organization. Not every subdivision has rich club organization. There are differences, and I think they relate in some ways to the way these systems are functions, what their primary roles are in the rat CNS, and their evolutionary history as well. One thing, a methodological point I want to make is the following. I've been talking about modules in a sort of informal way, as if they're easy to discover. They are actually not that easy to discover, and there's an entire field in network science of what's called community detection, which is essentially the principled way of defining and detecting network communities in large networks. This is a non-trivial exercise. And there's a methodological angle here that I want to press with you, and that is that I increasingly believe that modules exist on multiple scales. And in fact, to really characterize modular organization in a nervous system, we have to pay attention not just to one scale that is often chosen somewhat arbitrarily, but actually multiple scales at once. So here you see a connection matrix from the rat end brain, which is the union of the cerebral cortex and the basal ganglia, telencephalon, and we've clustered it here to show you what the major modules are, as it were. But if we actually look at all possible modules that we can detect at different levels of scale, we have a resolution parameter we can actually vary, sort of like a zoom lens on our detection algorithm, that allows us to pay attention to smaller or larger scales at times. And if we take all of these scales together and we form what's called an agreement or a co-classification matrix, that in turn can be hierarchically clustered and allows us to see the nested organization of modules in this subdivision of the rat brain. Each of the elements in this matrix corresponds to an affinity or probability, if you wish, or frequency, which refers to how stable that node pair is affiliated across multiple scales. And I've become very fond of these co-classification matrices. They are very nicely behaved. They range from 0 to 1. They have well-defined null models we can apply to adjudicate whether clusterings are above or below chance. And they provide us with a summary picture, a visual, if nothing else, a visualization of the complete community structure of a network that we're interested in. They also are, by the way, I'll give you a little tidbit here that this has not been published yet. They also quite closely related to functional connectivity. I'll come to this point. Maybe I'll come back to it a little later. So, bottom line here, methodological point. These days, in our lab, certainly, when we're doing community detection, we always go full, we look at all scales at once. There's no reason to believe that any one of these scales is particularly privileged over another. And we often try to summarize our observations in these co-classification matrices and then cluster this, which then becomes a hierarchical tree of nested modules that are sort of modules within each other. The latest iteration of this came out last year, and it's the Rat4 brain. It's a fairly big chunk of the rat brain, 466 brain regions in both hemispheres, comprising cortex, basal ganglia, thalamus, and hypothalamus. And we are now underway with the midbrain and hopefully have the brain at some point in a year or two, with probably 900 or close to 1,000 brain areas in the rat. In this latest endeavor with Larry, and I have no time to really go and dive into all the details, what has been impressive to me is that that these blades in the hierarchy that you see, which are nested modules, actually have, when viewed by a neuroanatomist who is familiar with rat brain anatomy and physiology and behavior, have sometimes strikingly clear behavioral connotation. We now talk in our own communications quite often about these modules as subsystems that have actually functional roles, distinct functional roles to play, and we can discover them with multi-scale, what we call multi-resolution consensus clustering, which is the tool that we apply to anatomical brain networks more and more. So the wonderful detail here, I'm just giving you a hint of it. There's much more in this paper and in the supplementary information, especially. It is something that to me is very rewarding. As someone who's worked on networks for a long time, I've always tried to, I've always hoped that this stuff is useful to real neuroscientists, real neurobiologists, and this is one instance where apparently insights can be gleaned from the types of analyses that we do on networks. Just one last point about comparative. Just one last point about this cost efficiency trade-off that I'm still quite fond of as an idea. Worked on this a long time ago now with Ed Bullmore. Why does the brain look the way it does? It's sort of a question that it tries to answer. We have complex topology. We have some local features like modules and high clustering. We have shortcuts or long distance connections that are costly in terms of wiring. That's the mixture of features that we find. And we believe, and I still believe that to be a valuable idea, that this represents kind of a compromise or a trade-off between two opposing tendencies. One, to make the brain as cheap as possible, make all connections as short as possible so that the least amount of wiring and energy is consumed. That, of course, leaves us with a terribly insular brain that is very difficult that has a hard time communicating with itself, especially between the outer edges. Random topology, on the other hand, is perfect for moving information around very quickly, except there is no information because everything is the same, and there's that sense of mixture, and specialization is lost. Here we have the two combined. This has to do also with an old measure of complexity that I'm still very fond of, also that we worked on with Julia Giannone and Gerald Edelman back 25 or more years ago on complexity being essentially the union of segregation and integration, a concept that I think is very useful. Very brief advertisement here, comparative connectomics, comparing connectomes in a principled manner between different species has now become possible for the first time. We have a rodent brain, we have avian brain, we have invertebrates, we've got non-human primates and humans. And we also have more than that. We have now, thanks to the efforts of collaborators that I'm starting to work with in Israel, a whole database of mammalian brains from animals that have been brought to the lab that have passed off natural causes in zoos or in the wild and have been scanned. So I have in my possession, in my prized possession, a leopard connectome, as well as the capybara, which is everyone's favorite rodent, that lives in South America. And we are engaged in a comparative analysis to see what kind of structural topological features we can see are changing in a systematic way across the mammalian class. Right. right about at the halfway point, which is where I wanted to be at this point. This is good. And now I'm going to pivot over to functional connectivity and dynamics. I've talked really only about anatomy so far, and structural connectivity, for me, a very important part of the equation, really the bedrock, I think, of neuroscience in some ways. Epitomized here with that image from the old study with Patrick Hagman. But another question, of course, is how do we get from something that is relatively sparse, static, almost like a skeleton on the left, to something which is fluid, dynamic, and ever-changing, functional connectivity and functional activation patterns, dynamics really on the right. And my lab, to this day, I'm obsessed with understanding how these two worlds interact with each other. This is probably, I'll work on this for as long as I can. I'm pretty sure there's lots still to be discovered. A couple of things I want to highlight here with you. First of all, what's functional connectivity? Many of you know this. We take a movie like this, resting state actually in this case, we cut it up into frames. The frames are individual pictures, you might say, of activation patterns, bold activations across the cortex, across the brain. We cross-correlate them. In most studies, Pearson correlation is still used as the default measure of functional connectivity. It's just simply a statement about the similarity of the ups and downs of the bold time course. Clustering a matrix like this gives us very quite reproducible, I would say, patterns, especially when carried out in larger cohorts. And these blobs that you see, these orange spots, which are highly internally coherent, assemblies of brain regions, these have now gotten names such as default mode network, control network, dorsal attention, saliency, ventral attention, somatomotor, visual, you name it. And we forget sometimes how this nomenclature and how this taxonomy of resting state networks has changed the way that we are describing the brain and how we are studying it in cognitive and systems neuroscience. Again, when I first got started in the field, there was no resting state, there were no networks, and there certainly weren't any resting state networks taken together. So this has all been quite a revolutionary over the last decade and a half. As you all know, large cohort surveys of functional connectivity taken by different imaging centers across different cohorts of subjects come up with fairly similar breakdowns of these networks in terms of major components of them. These are really nothing else but communities or modules that are being detected with various approaches in functional connectivity networks. So that's become a bread and butter set of terms and a way of approaching the brain that really didn't exist until fairly recently. In our own work, we've been interested in, for a long time now, in trying to build sort of generative models for functional connectivity. This line of work goes back, in my case, all the way working with Michael Breakspear, with good old Breaky, back in the day, implementing coupled differential equation models on coupling matrices. Here, the binary directed matrix coming from the macaque visual and somatomotor cortex that I've shown you earlier. In our very first set of studies carried out with Chris Honey, my former graduate student, with Breaky and with Wolf Cutter, we developed sort of a generative model for bold signals and bold signal fluctuations that occur spontaneously in such a network. And years later, actually, I got an email from Japan, from a lab in Tokyo, where they were interested in looking at comparing our model to empirical data they had recorded in monkey resting state anesthesia, actually, which you see here on the left side of this diamond-shaped figure at the bottom. On the right is our model that we had done years before. And while the comparison is certainly not perfect, but given that there was no data fitting at all, that's perhaps to be expected. It's still highly significant. So our model captures a certain amount of variance that's present in the actual macaque brain in terms of bold dynamics. A different line of work then in my lab was to make the model simpler. We wanted to come up with, do away with dynamics for a little while actually, and look at analytic descriptions of functional connectivity or predictions of functional connectivity coming directly from the structural connectivity matrix based on a combination of graph theoretic features. We did that, and my former postdoc, Joaquin Goni, is showing here on the left an empirical functional connectivity matrix, on the right a prediction of functional connectivity coming from the structural connectivity matrix and a variety of graph metrics without a whole lot of overfitting, I would say, just a few beta values. Again, highly significant agreement between these two domains, modeled and empirical. And it shows, I think, the potential success of these models, but also the fact that a lot of functional connectivity, not all of it, but some of it, is driven by underlying structural connections. Today, this does not seem surprising, but at the time, again, a dozen years ago, I don't think people thought of it that way. And I think these models continue to be useful in building intuition as to where functional connectivity comes from. A real shift in the last few years has been that even functional networks now are being viewed as more dynamic than we used to think of them. This is a little movie we made in 2007 of the macaque cortex and how with fluctuating bold signals projected on it coming from our simulation. And what we noticed in that paper, and we wrote about it in that paper, 2007, was that when we did windowing of functional connectivity with 30-second windows sliding across a 15-minute or so time span, we found that graph theoretic, the status of brain regions as hubs or non-hubs, for instance, fluctuated across time. And it did so without any active mechanism doing any switching or triggering a transition or anything like that. This is simply because a high-dimensional system of coupled differential equations can, under the right circumstances, give you additional timescales to the ones that you've built into the differential equations themselves. So here we have very slow fluctuations. These are seconds, time base here is seconds, very slow fluctuations of the status of a prefrontal area, for instance, in terms of whether or not it is highly connected or not highly connected within its own functional network. And this, to me, was a very important lesson. We actually, we tried talking about this to people, and there were two reactions. The neuroscientists were not interested, and the physicists thought it was all old hat because, of course, you get additional time scales when you have high-dimensional dynamic systems. This did not seem surprising to them at all. The work has been carried forward over the last decade or so with many studies, especially coming from some colleagues in the field here, Gustavo Decco, Victor Yerza, Randy McIntosh, etc. All of which combined kind of have shown us a lot about what goes on in functional connectivity, the dynamic functional connectivity, the relation of the dynamics we see to multistability, metastability, and near criticality. Our own work over the last few years at first focused on fluctuations in modularity over time and empirical data, working with my former postdoc, Makoto Fukushima. Makoto built a fairly robust way of detecting high or low modular episodes in functional connectivity, and then went to try to characterize what these patterns actually looked like and whether they were similar across subjects and across data cohorts. And he found that indeed high and low modularity episodes had some signatures to them that were stereotypic and reproducible across subjects and across imaging sites. High modularity episodes are characterized by a disconnection of what we might broadly call task positive from task negative regions and networks and systems in the human brain, while low modularity periods are much more flat in their appearance on the functional connectivity matrix and contain a much more heterogeneous set of states. We were very keen to look at the superb studies that Mac Shine carried out in during those same years and learned a lot from them. And we were kind of on a parallel track for a little while, and this sort of segregation, integration, fluctuation has I think underpinned some of our common work that we've carried out over the years, and we are still very keen on understanding what goes on here. So one upshot of Makoto's work was that This common sort of fingerprint of high modularity states was reproducible across different cohorts of individuals, and this fingerprint of default mode control on one side and attentional and sensory motor systems on the other side has stuck in my mind over the years. And in fact, we have now rediscovered it, as I'll come to in just a minute, in the latest work that we've carried out. Makoto has since then published a beautiful, I think, series of papers looking at where these fluctuations come from in simulation, in building neural mass and Kuramoto oscillators that can generate bold dynamics synthetically, and then looked at fluctuations there and try to trace what these fluctuations are caused by in terms of the underlying connectivity pattern. Now I come to the work that I'm really excited about. I want to talk a little bit about with you, and that's been triggered about a year and a half ago now, especially by my grad student Josh Faskowitz, who was going to graduate next month, get his PhD, and will be on the job market, I might add, and Rick Betzel, my colleague here at IU. These guys really get all the credit for the initial discovery of what we're going to be talking about here, and that is the following. Take a step back for a moment. Classic functional MRI functional connectivity is based on node activation patterns and Pearson correlation among them. This is the simplest case. I know that not every study is done that way, but let's just stay with this for the moment. So it's classic Pearson correlation among nodal activation patterns. What Rick and Josh proposed was edge functional connectivity based on a similarity of edge-based co-fluctuation patterns. What is that? Here's how we get to that construct. On the bottom here, we have two Bolt signals. This is standard stuff, right? The Bolt signal goes up, it goes down, et cetera, et cetera. We have z-scored here, we've normalized it. And the product of these two time series, the element-wise product of these two time series, the average of that is equal to the Pearson correlation, okay? So in other words, the element-wise product of these two time series, which is represented here, the average of all these points that you see here is exactly equal. It's not an approximation or whatever, it's a mathematical equality, is equal to functional connectivity, a single functional connectivity estimate of Pearson correlation. This triggered several ideas in our heads, especially in Josh's and Rick's heads at the time, and then they looped me in, and you know, the rest is history, as it were. This co-fluctuation time series is essentially unfolding on each edge, on each node pair separately, and can be treated as such. So what we can build is we can build an edge by time matrix, where the rows in this matrix are individual node pairs I, j, and the columns are time points. The mean of this matrix along the y-axis gives you a single vector, which when you fold it back into square form is exactly equal to functional connectivity. You can also go the other way. You can vectorize the upper triangle of an FC matrix, get this vector, and then unfold it, unwrap it in time, which is what you have here on the right. I want to stress that this is not an approximation. Nothing has been added here. This is a perfectly sort of organic way of dealing with the data. Nothing, no additives, no preservatives, no nothing. These components that you see here, this is an exact decomposition of what typically goes into functional connectivity estimates. Now we can take this edge by time matrix, and this is what Josh and Rick have been doing for the past year and a half. I have pounded on that a lot and got a paper out in Nature Neuroscience on it. We can actually look at a similarity of these time courses. So not the node time courses anymore, but the edge time courses. Gives us a much bigger matrix of node pair by node pair, and we call that edge functional connectivity. There's interesting, when you focus on edges and you start clustering edges, for example, interesting things happen. For instance, if you want to map this back onto nodes now very naturally, in fact, obligatorily, nodes have overlapping affiliations with edge communities. This must be so because of the nature of the clustering that you perform. This has been pointed out by people who have been doing this in network science for many years. So doing this and clustering forming edge communities essentially, and mapping them back onto the brain gives us a new way of looking at how different brain regions, for example, participate in different edge communities. The edge communities, you can think of them, I think of them as different conversations that go on in the brain because they are coherent and have high similarity amongst each other. There are similar time courses in the way both signals fluctuate across these edges. That's one way of thinking about it. And Josh and Rick have been spinning off many applications of this approach already in numerous papers in the pipeline. And I don't have time to go into edge functional connectivity in great detail today. Instead, I want to slow down a little and pursue a parallel track that I've been particularly fascinated by, and that has more to do with the moment-to-moment fluctuations that we can observe when we look at edge time series. So once again, this is our picture. We looked at this before. And perhaps you've noticed something. There are these vertical streaks going through this edge time matrix. This was very worrying to us initially early on because we live when you live with fMRI data, you always worry about artifact. That's what you do. That's what we do every day. We worry about artifacts and usually very unwanted artifacts such as head motion or scanner noise or who knows what else. Things we haven't even thought of yet that are suddenly appearing as artifacts in our data. We tried all sorts of things, trying to track down what potential artifact could cause these vertical streaks around the edge time matrix and came up with nothing. Nothing seems to covary particularly strongly or at all, especially not head motion, with what we're seeing here. Okay, so these we can sort of say that whatever the brain is doing, perhaps these brief excursions of what turns out to be high edge co-fluctuations on a brief interval may not be artifactual, but may actually be something the brain is doing. So if we plot a summary statistic across time of these edge time constructs, we call it the root mean square or the root sum square really of the amplitudes of the edge co-fluctuations. We're getting this time series below, and we can then detect events. We call them events because they are surprisingly high co-fluctuation occurrences across time. Notice here the time base. This is a 12 minute run, and these events don't occur that often. Okay, so they're quite infrequent. So another thing I want to point out is that whatever goes into this, so the construct on the left is what we're all familiar with, right? Functional connectivity matrices, Pearson correlations of old time courses. I want to point out again that if you don't like these things and you think they are somehow garbage or whatever, they go into this, what's on the left, okay? And in fact, not only that, but they drive what you see on the left. Because they have high amplitude, they're actually factoring into the average estimate of the functional activity quite a bit. So whatever it is we're pointing out that's going on here, it is in our data. And it is, we have looked at lots of different data cohorts, and we consistently find these events across scanners, across processing algorithms, et cetera, et cetera. So edge time series exhibit bursty behavior. These, we call them informally events. These are unrelated to things that we don't care about, and they are intermittent, short-lasting. They are present in movie data. Interestingly, in movie data, they line up across individuals. This is something that we are planning to dig into as soon as we can take a breath and actually get to it. They can be extracted also from standard bold time series, and they're present really in all individuals we have ever looked at. They are ubiquitous in data of this type. So what does classic functional connectivity look like around the events? For that, what we do is we shuffle our individual time frames, and we order them by signal amplitude. We order them high to low or low to high, and then we build we gradually include more and more frames in our average, and we build a functional connectivity matrix going from high to low or from low to high, from event to non-event or from non-event to event. When we do that, we approximate the final outcome, which must be up here at the intersection with 1 at very different speeds. If we go from events to non-events, even a small sample of our data, 10%, 20% of our frames, time frames, the high amplitude time frames, approximate the final pattern very well. This is not the case when we go the other direction and start sampling those low amplitude frames first. We are getting a much less precise, much slower approximation of the final pattern. So this is interesting. It tells us something about what these event frames are doing. They're driving classic functional connectivity, and hence they're also driving classic system level architecture, resting state networks, which are enshrined in our understanding of functional connectivity. These features that we are all familiar with, they are probably driven by these event frames, by these spikes in high amplitude signals that we observe. The non-event frames, on the other hand, are not driving the final pattern nearly to the same extent. In previous language, event frames would correspond to what we might call highly modular time points, and non-event frames might correspond to low modular time points. I didn't bring the figure with me, but in fact, modularity is quite different between these two types of functional connectivity subpatterns. When we look at the pattern that we actually, when we try to map this back onto the brain surface doing PCA, we get a principal component that, when mapped onto the 200 nodes that go into the analysis, looks like this, and it in fact corresponds quite closely to control and default mode regions on one side and everybody else on the other side, by and large. And that is, of course, the pattern I pointed out to you earlier that we had looked at in more classic windowed analysis of time of functional connectivity patterns, where the high modular patterns had that particular signature to it, refining it here at a single frame resolution. I'll show you a movie, why not? Here's a movie of a single fMRI run from the Human Connectome Project dataset, 1 subject, 1 run, 13 minutes. What you're seeing is at the top in the square array is the edge co-fluctuation pattern mapped back onto node by node space for each edge. And you're seeing it across time. So this is literally a movie of every frame in the run as it unfolds. The mean of what you're seeing up here in that 200 by 200 node array is exactly equal to the functional connectivity, okay? We've been over this before. What you see below is our edge by time matrix gradually filling in as we're traveling through time. We're seeing these events occur, peaks that have particular shape and particular topography to it. What you're seeing here is the 7 canonical systems. So the system has been organized in this manner, and I would like to just point out to you a simple observation, eyewitness account almost, that the individual functional systems that you see listed here do not really manifest all that well most of the time. We saw some automotor pop up in a bright red splotch. This meant all of its regions were co-fluctuating, but that was a brief instant. A lot of the time, the pattern looks fairly disorganized, and only occasionally do we see manifestations of individual systems that pop into existence and then disappear, or combinations of such systems, as you just saw. Here's some automotive system again. So again, what the observations that you see at the top are the raw material that go into functional connectivity. We've not added anything. This is, we have not processed the data any further. There's no fancy stuff going on here. This is simply frame by frame the bull time series, and the co-fluctuation being the product, element-wise product of the bull time series on each edge. Now, when you look at something like this, you, at least me, want to do some MATLABing, right? And what we one direction that we've taken this is I've been interested in decomposing these arrays further into binary patterns. Binary is nice because it's compact, it's simple, and it can be used for all sorts of applications down the road. Here's our full analog, if you wish, edge time series by frame matrix, which again, if we average along the y-axis, gives us the exact functional connectivity. What we discovered is it's a simple mathematical exercise, logical exercise, that on each time frame, the co-fluctuating sets that are present in the brain can only be 2, and exactly 2. So there's a bipartition at each point in time if you're staying with the edge co-fluctuation framework. These bipartitions, when we superimpose them, form a so-called co-classification or agreement matrix, are almost identical to the functional connectivity. The correlation is something like 0.96, 0.97, something ridiculous like that. So this binary pattern down here of bipartitions, of the sectioning the brain into 2 sets of internally co-fluctuating but otherwise opposed sets of nodes on each time step, this binary decomposition gives you back, if you superimpose all the bipartitions, almost exactly gives you back the functional connectivity pattern that you get from the analog. I call it the analog version of the data where the amplitudes have been preserved. There are some interesting little games we can play with that. For instance, we can take these are binary patterns, so we can actually cluster them, classify them very handily. Binary patterns are great that way, and we can, for instance, classify them as each one of them as a member of a class that is defined by the various subdivisions of functional systems we can perform. So if we have 7 functional systems, it turns out we can have 63 different types of bipartitions that we could encounter. Simple combinatorics. We can now ask for each point in time, what is the closest that each frame gets to one of these templates up here? And then we can classify that frame accordingly. So it's like doing a clustering, but where the clusters are predefined by a prior knowledge you have about what you expect to see, which is in this case functional systems. When you look at these types of analyses, it becomes apparent that functional systems, visual, somato-motor, default mode, etc., are not continuously expressed. In some ways, do not exist continuously through time, but are expressed only transiently and intermittently. And we can pinpoint when they are expressed precisely with single TR resolution. You saw the movie, right? And here's a time series of the corresponding mutual information values of some of these templates with actual empirical recorded frames. So the long time average FC that we're so familiar with, that so many people have invested so much into, is really a superposition of many short-lasting episodes, many bipartitions, during which functional systems aren't usually well expressed. But if we superimpose them and we sort of average across, we get the classic functional architecture we're so familiar with resting state networks and the like. This approach opens up a whole Pandora's box, I'd like to say, of possibilities. Things we can do with binary patterns. We can cluster them. We can compare them across individuals. We can look at when they're expressed very highly and when not. We can look in tasks. We can look in movies. We've done some of those things already, but there's much more to be done. And I would be very eager to get some of you interested to look at this approach. We have a new paper out here, and there's code attached to this. The basic constructs to do this are, I would say, really quite simple. And again, do not require elaborate computation or inferences or statistical operations, in my opinion. One direction we've taken, this is an upcoming OHBM presentation here by my graduate students, Sarah Katz. We have looked at up here once again the full pattern of edge by time and the functional connectivity, and then we look at how well do these individual frames in the fMRI time series, how well do they represent particular templates of the kind I just showed you, those 63 subdivisions of functional systems, 7 functional systems into bipartition sets. And we can quantify how well they approximate visual information, and then we can stratify our frames, order them by magnitude of that metric, and then we can do something fun and simple. We simply select the top 10% of those. So we're going to pick out of these time series just those points when the individual frames were particularly well aligned with particular templates that we've provided. And then we can take these 10% of frames and average them together. This gives us a functional connectivity component. Strictly speaking, not functional connectivity because we're not renormalizing our time series, but it's a component that has bearing on and often resembles what we see in full functional connectivity. So this is a component of edge co-fluctuations over a period of 10% of the frames under a particular criterion of filtering, which in this case is a template that selects for the ventral attention and the frontoparietal network to be highly expressed, and the rest of the brain being anti-expressed, if you wish. That is our bipartition. And we can then ask questions about identifiability. Now, this is, of course, the classic work by Finn et al. Looking at, how well can we identify subjects across individual imaging sessions? And we find that with some of the templates, but not all, we can get really good identifiability. So we're thinking of this as tapping into a particular source of individual differences, not on the rows of that matrix at the top, but on the columns, which is time. And finding that different templates that we throw at this analysis give us different levels of identifiability, which tells us that identifiability of the characteristics of an individual's pattern that make that person's pattern fluctuate over time. So cortical fingerprinting, as it's sometimes called, or brain fingerprinting, not only has a spatial signature with some systems privileged, frontoparietal and attention systems, for example, but it also has a temporal signature. In other words, those differences don't manifest equally across all time points, but fluctuate. Coming to the end, I'm almost at the end, wrapping up a little bit. There's many other things I could have talked about, communication models that I'm very keen to get back into, etc. But a few words about communication and how it might link together structure and function in brain networks. This is the way I think about it anyway. On the left, we have anatomy on sort of our foundation. The structure connectivity for me is important because it is the bedrock, I think, on which everything else unfolds. And within the anatomy, we have signaling events that must occur on structural pathways, right? Think of these as action potentials, messages being relayed from brain regions to brain regions, etc. On the right, we have functional connectivity, which is the combined total, the outcome of all these communication events. So functional connectivity is not communication. It's difficult to infer communication from just observing time courses, right? I mean, this is a classic problem of inferring, hidden causes that you can't actually see directly from the consequences that you observe, which are the statistical dependencies among the ups and downs of a bull signal, let's say. So we have, I think, good data here. We have good data in the anatomy. I think it's pretty good. What we're really lacking is an account, even just a way of observing what goes on in the middle. The ebb and flow of signaling events in the brain is not directly accessible to us, I would argue. We are seeing consequences, we're seeing time courses, yes, but they are not the communication events themselves. And this is, to me, a sort of a hidden a treasure trove, potentially, of insight, if only we had proper methodology, both empirically and theoretically, to get at the ebb and flow of communication dynamics, of signals being exchanged at a very high rate and I'm sure, very dynamic patterns across time and space. I've gone through all these topics with you. I'll end with saying that network neuroscience as a field is now fairly well established, and I have in my spare time started a journal, open access, that is published by MIT Press. Now it's in its 5th year of publication, and we'll soon get an impact factor. So we are very eager to see what that might be. And I recommend it to you if you have work in this area. Thank you very much for your attention. I'll unshare my screen, and I'm sure there are some questions that have occurred to you.
Everyone's muted. I'm going to clap for everyone, Olaf. That was absolutely fantastic talk. So if anyone has questions, please share them in the chat, and then we'll unmute you and maybe get you to put your screen on so we can ask some questions. We've probably got time for about 3 or 4 questions. In the meantime, I might take the host's privilege. So that, I mean, you covered so much ground there, Olaf. I think I could ask you questions about 20 different parts of your talk. And you can probably tell that my neck looks a bit stiff because I've been vigorously nodding along with so much of what you've been saying. One thing that I kind of wanted to kind of get your take on a little is whether or not the concept of the network that we're mapping at the moment, it really is a match for the brain, or rather for the cerebral cortex. Right? Your work with Larry Swanson will testify that there's so much complexity in the subcortical structures of the brain, how they interact with one another. And they often do so in really kind of interesting and constrained ways that are quite different than the kind of pattern we see in the cerebral cortex, where there's a lot of kind of interconnection between nearby regions. And in the subcortex, you can have very tight distinctions between very different subnuclei that have completely different projections. And so I wonder whether you've got any sort of thoughts about how we can embrace that perspective and think about the subcortex, and what kind of influence that might have on the sort of things that we've been measuring in the cortex, like rich clubs and you know, integration segregation balance.
There's several questions rolled into one here. Number one, because of the richness of the biological domain in general, I think, but brain in particular, I think network models and the framework of network science is not in itself just the only thing that we need. Obviously, we need other things too. We need evolution, we need development, we need all sorts of things to come together to understand how the brain works. Networks aren't everything. However, I do think they apply, but in different ways, to different anatomical subdivisions. So one upshot from the work with Larry has been surprising to me, quite frankly. We started in the cortex and found all the things that we're so familiar with, the modules and the rich clubs and hub nodes and all of this good stuff. But when you look at other subdivisions of the brain, hypothalamus to me was a real eye-opener. It's a much smaller structure, much closer to the midline in terms of where it's positioned, and much older. And as you point out, with a lot of very intricate anatomy and physiology on top of that, different cell types intermingled quite tightly, and anatomical subdivisions. A Rubik's Cube in there, right? It's sort of like it's all folded into. It's like something you bake in an oven. You fold the dough again, and it becomes this enormous mess. And so, for instance, that structure does not show small world properties. It does not have any obvious resemblance to cortex along many of the classic dimensions I mentioned so much today. So we should, in fact, be quite open to the realization that other parts of the brain are not like cortex and are not perhaps optimized for information integration. I think hypothalamus has very different priorities than that. And it's also a much smaller structure where, things like wiring costs don't play such an important role as they do in a very large, essentially a sheet such as the cortex. So absolutely the case, different types of architectures will be encountered, and we will encounter them as we dig deeper into it. On top of that, one thing I have not mentioned is the heterogeneity of cell types, of just different morphologies and different neurotransmitter systems and different gene expression patterns that we have to imagine has to be layered onto a very simple adjacency matrix of the kind I showed you. In the current work with Larry, for instance, we have not yet gone into that domain very far, in part because the data is not there yet to the extent. But I think that will ultimately make our networks much more multi-layered, multiplexed, and will add a dimension to it that currently we don't really have. So So I'm very eager to join the post-network era when all of these things come together and,
I love it.
and will inform our understanding of the brain in a much more comprehensive way. Networks aren't everything, but I'd like to point out there was a time when we didn't have them, and it was a very cold, dark period. And now we've learned a few things from the studies that we've undertaken in the last couple of decades.
Yeah, no, great answer. I'm glad you brought up the cell specificity as well, because that was another one I wanted to pick your brain on. I think once we get things like the Allen Brain Atlas, where we can go in and develop Cree lines of individual neurons, and we can start to ask, what does their network look like, and how does it interact with another subnetwork? I think that's really, really exciting.
I'm pretty sure we'll discover new rules. Sort of, but in some ways we have to rethink our network approaches. They can't be as simple anymore as just adjacency matrices or even functional connectivity or whatever. It has to be much more multi-layered, multi-tiered, multi-omic. In some ways.
Yeah, 100% agree. I noticed that Eli Kornblath has his hand up and he's got his video on. Go for it, Eli. Whoop, you're muted, I think. Someone sec, mate. Can't hear you. Can anyone hear Eli? Olaf, can you hear Eli or not?
No, I cannot.
If you want to type your question into the chat, I can read it out for everyone. Hold on, have we got him yet? Ben and Joe, do you know whether or not you can unmute Eli?
He's not muted it. It seems like the mic's just not working.
Ah, got it. Can anyone lip read? No. Okay. Yeah, okay, here we go. He asked for a 1 second. Okay, no worries. I'll come back. Okay, hold on. So we've got a question. What's the advantage of edge-centric analysis compared to traditional node-centric analysis? Do you want to?
That's a good question. In fact, we've debated this in the lab also, and with Rick and with Josh especially. So one thing I certainly would like to point out that from a graph theoretic perspective, flipping the network inside out and taking the edges to be your nodes and the nodes to be your edges is possible. So, so-called line graphs have been around for a long time. This is a graph theoretic concept that, has a lot of theoretical work behind it. It just hasn't caught on in neuroscience for some reason, because we do think, I think, intuitively, that the nodes are doing the computation and the edges are just relaying the outputs of the computations, right? This is sort of a classic, in some ways, connectionist way of looking at the brain. And we think there's value in turning the model on its head and saying, let's prioritize the edges and let's think of them as conduits of communication, and let's think of the communication patterns themselves as the critical entities that we want to map out and cluster and display and explore. And that's kind of where we're coming from. We're just turning the model inside out. Rick would be a much better person to answer this question, because I think he has already provided in the published papers, a few of them are out now and others are snaking their way through the review, various aspects of what edges add to the picture, additional aspects of individual differences, for instance. Etc. In my own work, I have not gone to the edge functional connectivity side of things too much, in part because I was getting really drawn in by this frame-wise decomposition that I went on about at the end, which is where I've spent most of my own work. As Mac knows, I do my own coding. There's not a day that goes by when I'm not coding, and quite a bit actually, usually. And so I've been digging into that side of things myself quite extensively. I'm wondering what people think about it. Mac, we had a brief discussion the other day, and you were pointing out to me that when you go to these high frequencies, these fine sampling rates, there's a trade-off because you're getting a lot of noise and potential statistical issues. To which I say, I haven't done anything to the data at all. I just display it. I just unwrapped it. So everything that you see in that edge by time matrix goes into what what then goes into what's functional connectivity. So if there is issues with the frames, then let's address them, because it's also coloring what we see at the long time scale, right? So this is my defense here, is that we have not done we're not doing windowing. In fact, windowing to me is now a thing of the past, quite frankly. And we're not doing any processing really on top of what we're measuring. We're just looking at it in the sort of natural state before it becomes a correlation.
Yeah, what you see is what you get. I guess the question for us to kind of work out as a field is, which elements of those parts of the data are ones that are associated with something like an underlying neural event versus something that maybe something that we're not interested in. And I'm on the side of you. I prefer to see all the data and then try to work out post hoc what could have been caused by some event of non-interest, rather than sort of removing everything and then only looking at the residuals. That always makes me a little uncertain. So let me just quickly jump. It looks like we might have Eli back now. Eli, do we have a microphone?
Hey, can you guys hear me now?
Yep.
Yes.
Awesome. Okay, thank you so much, Dr. Sporns. That was an excellent talk. It was really cool to see you walk through everything from structure to function. And thank you very much, Dr. Shine, for hosting this. The question that I have is are there methods that you're aware of that can take a more spatio-temporal approach? Because I think sometimes when you look, for instance, when you do community detection or unwrap these edges and look at them as individual time points, you lose something like a wave that might be traveling across the brain. I think that's apparent when you look at these videos of old activity on the cortex. You can see there's like a spatial temporal continuity to the changing activity. And I'm wondering if there's any approaches that could account for that.
Well, it's wonderful you're mentioning waves. I am, if I had a little bit more time on my hands, I would be all over that already. We are seeing evidence of what is exactly what you sort of see impressionistically in the movie. We're also seeing it statistically. There are waves that wash over the brain, as it were. The point is that with this little trick that we introduced a year ago or so, with this frame-wise decomposition, we can now see these waves, and we have now not been able to see these really before at this level of detail, and some of the things that I mentioned allow us to sort of analyze them in a more rigorous way with statistical tools and so forth. So yeah, absolutely. I might say that these events that you see, they are somewhat more stereotypic. They're more alike with each other than other randomly sampled frames would be. So there's something about the brain recurring when it does that to some common state space, if you wish. But they're not all perfectly alike. So there is now some evidence that Rick has been very excited about on, looking at different types of events, different classes of events, etc. I'll very cautiously say that I have spent. In fact, I'm on my workstation that sits next to me right now. I'm running simulations of neural mass models and core motor oscillators, for instance. And we're seeing event-like patterns in those simulations as well. This is to me compelling, because we cannot say there's no head motion, there's no respiration, there's no scanner artifact. It's just coupled differential equations, and we see very much the same phenomenology using the exact same analytics that we're using in the empirical data, and we have an idea as to where it's coming from. I will tease with that. I won't disclose what it is, in part because I'm really not sure yet. But it's an idea that we're pursuing right now. We feel like we're hot in pursuit of what the actual causal mechanism might be that is causing these events to occur, at least in the simulations. I mean, not the real brain. It's everybody's guess, but. So these are not totally obscure things. I mean, once you get something in a very simple differential equation model, you sort of say to yourself, well, at least there's a simple way to generate them. It's not something that requires a lot of, extraordinary manipulation or weird fMRI, funkology, which we're all familiar with, right? So it's coming out at least in that domain as well, and that's to me very exciting. Again, it makes me feel young again when we were working on with Reiki back in the day on these first sort of oscillator models of bold connectivity.
Yeah, excellent. So apologies to all of the amazing questions that are coming in on the chat, but I think we've only really got time for one more. Sorry again, Stephanie, I just saw you put your hand up as well. So we might make Lorenzo the last question, but if you do have burning questions, maybe we can continue on Twitter, or people can email separately and we can forward things on to Olaf. I just want to make sure that we're not, taking everyone's time today. So Lorenzo, thanks very much.
Thanks for the privilege of picking the last question and for the great presentation. I wanted to go back to one of the previous question about the omics questions in the brain. And one of the omics that I've been obsessing with is actually autonomic physiology, respiration, heartbeat, skin conducts, and it's exactly those signals that we sometimes would like to, take out from the brain. And I was just curious to hear now your take on about, now that we're getting more and more nuanced measures of brain states in time, whether you think we could get closer also to understand which aspects of this physiology are just, noise and which are contributing actually to different brain states in terms of cognitive brain states like emotional states or different cognitive states. Just a curiosity about how these different signals could actually be contributing to some of the patterns you see in a meaningful way.
Yeah, it's a great question, and as I was presenting my talk, I took the vantage point of the person who really doesn't want any of that physiology. I mean, most practitioners in MRI do not really want to have a signal in there that's related to heartbeat or to some autonomic fluctuation or some other physiology that's considered to be a nuisance variable. However, having said that, I totally agree with you that these domains are important and probably underappreciated in the way that they impact and interact with brain function and even brain functional connectivity. I have personally no problem thinking of brain and body having a dialogue, for instance, the brain being just one organ amongst others and sending, and receiving signals from virtually every tissue in the body, having some responsiveness to that, and also perhaps even talking back to the body in some ways. I have no problem with that. I know very little about it, I'll be honest with you. I'm cheering on the efforts that are underway to unravel those relationships because I do think intuitively I feel like those are important. And perhaps these frame-wise decompositions could contribute to understanding better what goes on, because you can now track fluctuations in not only bold signals but also co-fluctuation patterns on edges at the time resolution of about 1 second, and that might be helpful in doing these kinds of analyses and seeing greater clarity emerge.
Thanks a lot. That's super interesting.
Hey there. Yeah, I just got the invitation to unmute, so I guess we have time for another question. Is that true, Mac?
Go ahead.
Awesome, thanks. So fantastic talk. So I'm really intrigued by the selection of time points based on the Edge time series before the performing of the identification. And I'm curious to hear your perspective a little bit more on the balance between selecting those time points that may carry more signal, maybe like in a denoising kind of fashion, versus just losing information, because we know that the more information that you have in the time series, the better the fingerprinting, better reliability, all that stuff. I'm wondering, have you specifically looked at this, whether you start to see this kind of inverted U relationship as you increase the threshold? At least I know right now you're doing this based on similarity with the frontoparietal and attention networks or attention template. I'm curious whether you started to see this kind of relationship.
We have not looked at that that particular aspect of it. So you're absolutely right. Usually more data is better, right? And especially for fingerprinting, this has been shown. However, in this case, my hunch is think this is kind of sort of direction we're taking with this study, which we're hoping to be writing up over the summer, hopefully, that sometimes less information is actually better. Why? Because if it is true that in the sort of signatures of individual differences are expressed in a, not in a constant way across all time points evenly, but are expressed in a sort of punctate fashion, then identifying where those points occur and removing the obscuring frames, the frames that actually don't contribute or perhaps obscure individual differences, might actually give you a boost. So less data may be more in this case. It's my hunch. And in fact, we've already shown that we can at least with the sort of standard measures of identifiability, accuracy, and a few others, that we can get pretty good performance on 10% of the data under particular criteria that are mathematically specified. So it's not just picking things at random. It's things that have to do with, you know, what do the patterns look like, which systems are involved, and the antagonism in the bipartition framework. So, I that's the direction we're taking right now. You know, it's thinking more in the direction of how can we unveil perhaps previously hidden sources of individual differences that have been obscured by the majority of time points that don't contribute much to individual differences of the kind that perhaps we're interested in. It opens up whole new ways of analyzing data, by the way, of course. You know, we have not yet stepped into the clinical domain, but I'm very eager to look back at using these criteria that I've outlined to stratify individuals based on their clinical status, maybe, or look for relationships of some metrics we get from these functional connectivity components with behavior, with cognition, or with outcome. You know, again, there aren't enough time and there's enough hours in a day to do it all, but I want to encourage those of you who, if you have a few minutes or half an hour of your time and grab some of that MATLAB code that's now online, that gives you the basic constructs, the edge time series, the root sum square metric, and perform some basic things on that, you can apply this to your own data within minutes. It's super simple. And explore and see whether you come up with new angles on the story that we haven't thought of yet. You know, so it's more like, you know, I feel like a door has been opened here. I've been working with windowed, you know, functional connectivity for some years. I've been frustrated because these windows are terribly blurry. They are 1 minute or quite often a minute long. That's not a cognitively relevant time scale, okay? I mean, from the point of view of what really goes on blow by blow, moment by moment. And it's been frustrating for me. So now we can actually look at something. We have to be sure what we're looking at is not totally confounded, of course, but we can look at something at a much faster time scale in data we already have. There's no need to acquire anything differently. You already have bold time series on your desktop right now. And with the simple approach that I've just mentioned, you know, you can, in a half an hour, you can reproduce the main constructs I showed you today in my talk.
That's absolutely true. Really great jumping off point for a lot of different investigations.
Yeah, exactly.
So thank you.
So get coding out there, get coding.
Okay, thanks for the question, Stephanie. So Max dropped off the line, unfortunately. He's having some technical problems.
I made it. I'm back.
Oh, he's back.
Yep, go ahead, Joe. All good.
Okay, so I was just going to do the finish up and say thanks again to Olaf. I'll give you the round of applause.
Thank you.
Since we can't hear everyone else. So thank you very much for that great talk. To everyone else, thanks for coming. Thanks for the questions. I'm sorry, we really should probably cut it off there. I know there's more on the chat. Perhaps if Olaf's receptive to that, you can mail him your questions if that's okay. But otherwise, thank you very much for coming. As Max said at the start, we've got other talks coming up in the series, so keep your eyes out. So thanks again to Olaf. I'm going to end the recording on that note.